Hi students, today we will see about bioenergetic. What mean by bioenergetic? Bioenergetic is the branch of biochemistry that focuses on how cell transformed energy of them by producing, storing or consuming adenosine triphosphate. Bioenergetic processes such as cellular respiration or photosynthesis are essential to most aspects of cellular metabolism, therefore to life itself. First we are all going to what is bioenergetic? The study of energy in living system and the organism that utilize them. Energy required by all organism. It may be divided into two types, kinetic energy or potential energy. First we are all going to kinetic energy. What mean by kinetic energy? Kinetic energy means it is the energy of movement. The molecules produced energy, for example heat and light energy. And next we are all going to potential energy. Potential energy or stored energy is more important in the study of biological or chemical system which can be used to do work. The mechanical energy that a body has by virtue of its position, stored energy. And next we are all going to bioenergetic. Bioenergetic is the part of biochemistry concerned with the energy involved in making and breaking of chemical bond in the molecules found in the biological organism. Growth, development and metabolism are some of the central phenomena in the study of biological organism. The role of energy is fundamental to such biological processes. The ability to harness energy from a variety of metabolic pathway is a property of all living organism. Life is dependent on energy transformation. Living organisms survive because of exchange of energy within and or without. Bioenergetics, in a living organism, chemical bond are broken and made as part of the exchange and transformation of energy. Energy is available for work or for other processes, and then when weak bond are broken and a stronger bond are made. The production of stronger bond allow release of usable energy. And next we are all going to, it is the quantitative study of the energy transactions that occur in living cells and of nature and functions of chemical processes underlying this transaction. And next we are all going to law of thermodynamics. The law of thermodynamics or important unifying principle of biology. This principle govern the chemical processes, that is metabolism, in all biological organism. And next we are all going to the first law of thermodynamics, also known as the law of conservation of energy, states that energy can neither be created nor destroyed. It may change from one form to another, but the energy in a closed system remaining constant. In any physical or chemical changes, the total amount of energy in the universe remaining constant, although the form of energy may change. And next we are all going to the second law of thermodynamics. The status that when energy is transferred, the law of entropy. What mean by entropy? Entropy means it is just the measure of how ordered or organized some system is. That is called entropy. There will be less energy available at the end of the transfer process then at the beginning due to entropy, which is the measure of disorder in a closed system. All of the available energy will not be useful to the organism. Entropy increases as energy is transferred. In all natural processes, the entropy of the universe increases. And next we are all going to law of thermodynamics. We are already see about the law, first law and the second law. And next we are all going to free energy changes. It is useful energy gives change in free energy delta G is that portions of the total energy change in a system available for doing work. It is also known as the chemical potential, that is delta G equal to delta H minus T delta S. What mean by delta G? Delta G is a free energy of any moment, and then delta H means the change in enthalpy of the system in a reaction. And what mean by delta S? Delta S is a entropy, and T means it is absolute temperature. We have to mention Kelvin. And what mean by enthalpy? Enthalpy means thermodynamic quantity equivalent to the total heat content of a system, that is equal to internal energy. And next we are all going to types of reactions. That it is divided into two types. First one is exergonic and entragonic. In exergonic is a spontaneous reactions that releases energy. If the free energy change is negative, this reaction is due to loss of energy from reactant. So it is called exergonic. For example, catabolic reaction. In entragonic is an anabolic reaction. What mean by anabolism? Anabolism means it contrasts to catabolic reactions. Anabolic reactions involve the joining of smaller molecules into large one. The anabolic reactions combine monosaccharides to form polysaccharide, fatty acid to form triglycerides, amino acid to form protein, and nucleotides to form nucleic acid. The entragonic is an anabolic reaction that consumes energy. If the free energy change is positive, the reaction is called entragonic. For example, synthetic reactions. But all equilibrium it is 0. The energy coupling occur by coupling of exergonic and entragonic reactions and liberations of heat. And next we are all going to energy currency of the cell. ATP is the primary and universal carrier of chemical energy in the cell. The ATP converted into ADP plus PI. We have to get 7.3 kilocalories per molecules. And next we are all going to high energy phosphate. The phosphate compounds whose delta G value higher than that of ATP, they are called a high energy phosphate. For example, phosphoenol pyruvate, creatine phosphate. The phosphate whose delta G value lower than that of ATP or low energy compounds. For example, ADP glucose 1-phosphate. And next we are all going to phosphagens. They are storage form of high energy phosphate. For example, creatine phosphate in vertebrate muscles and arginine phosphate in invertebrate muscles. The ATP combined with the creatine that converted into ADP plus creatine phosphate. And next we are all going to adenylyl kinase. It is otherwise called myokinase. The 2-ADP converted into AMP, adenosine monophosphate plus adenosine triphosphate. When ADP is depleted, the AMP act a metabolic signal to increases catabolic pathway. The ADP can be formed from AMP by this reaction. And next we are all going to nucleosides diphosphate kinase. the ATP plus UDP. UDP means uridine diphosphate converted into ADP plus UTP. By this reaction, nucleosides diphosphate can form corresponding triphosphate. And next we are all going to oxidation reduction. That is called oxidoreduction. Oxidation, it is defined as the removal of electrons, and reduction means it is defined as the addition of electron. For example, Fe2 plus ion is oxidized to form what? Yes, Fe3 plus ion, that is removal of electron, and then Fe3 plus ion is reduced to form Fe2 plus ion, that is electron added process. The affinity of an oxidation reduction system of electrons is referred to as oxidation reduction potential or redox potential. And next we are all going to redox couple. A biological system which has a stored tendency to donate electron has a negative redox potential. The redox potential of a system is usually compared against the potential of hydrogen electrode at pH 7 with 0.42 volt in biological system. For example, redox potential 0.32. The redox system NADH or NAD plus. The electron flow from electronegative redox couple to more electron positive system. And next we are all going to oxidoreductases. Oxidation that occurs in biological system is called biological oxidation. The enzymes involved in oxidations and reduction reactions are called oxidoreductases. There are 4 groups of oxidoreductases. First one is oxidases, and second one is dehydrogenases, and third hydroperoxidases, and fourth oxygenases. First we are all going to oxidases. They use oxygen as hydrogen acceptor. For example, cytochrome oxidase, and then water forming. The H2O2, that is hydrogen peroxide forming. D-amino acid oxidase. The D means dextrorotatory amino acid oxidase, that is FAD linked, and L-amino acid oxidase. L means levorotatory amino acid oxidase, that is FMN linked. The xanthine oxidase, that is molybdenum containing, and then aldehyde dehydrogenase FAD linked. Next we are all going to dehydrogenases. It involved in transfer of hydrogen from one substrate to another in a coupled oxidation reduction reaction. For example, nicodinamide coenzymes NAD is involved in oxidation reactions. NADP is involved in reductive synthesis, and riboflavin coenzyme, that is FMN and FAD. Flavin mononucleotides and flavoadenine dinucleotide. And next cytochromes except cytochrome oxidase. And next we are all going to hydroperoxidases. They use hydrogen peroxide or an organic peroxide as substrate. For example, catalase and peroxidase. Peroxisomes are rich in this enzyme. The hydrogen peroxide combined with H2O2, same hydrogen peroxide, then converted into 2 water molecule. The catalytic activity for the enzyme catalase. And next we are all going to oxygenase. Oxygenase, they catalyze the direct transfer and incorporation of oxygen into a substrate molecules. The first one is dioxygenases. It incorporates both atoms of oxygen molecules into the substrate. For example, homogeneized dioxygenase, then tryptophan dioxygenase. It is a pyrrolase enzyme, and then monooxygenases or mixed functions oxidases, and then incorporated only one atom of molecular oxygen into the substrate. For example, cytochrome P450 and then cytochrome P5. Next we are all going to SOD. What mean by SOD? Superoxide dismutase protect aerobic organism against oxygen toxicity due to superoxide anion free radicals, that is O2 plus O2, that is oxygen compound plus combined with 2 hydrogen then converted into hydrogen peroxide plus oxygen molecules, then catalase removes H2O2. And next we are all going to oxidative phosphorylation. What mean by oxidative phosphorylation? It is the process by which free energy liberated when protons are pumped by respiratory chain is trapped in the form of ATP by phosphorylation of ADP with phosphate. The process of oxidation coupled with phosphorylation is called oxidative phosphorylation, and it occurs in the mitochondria. And next we are all going to most of the energy liberated during the oxidations of carbohydrates, fatty acids and amino acid is made available within mitochondria as reducing equivalent. The reducing equivalent thus obtained is oxidized by oxygen forming water, and the energy liberated is used for ATP synthesis. Mitochondrial inner membrane contain the respiratory or electron transport chain that collect and transport reducing equivalents. And next we are all going to respiratory chain. The components of respiratory chain are arranged in order to increasing redox potential, that is for NAD or NADH to water and oxygen. The redox couples components of respiratory chain. There are 4 complexes. First one is NADH, ubiquinone oxidoreductase, and complex 2 succinate ubiquinone oxidoreductase. Complex 3 ubiquinol cytochrome C oxidoreductase. Complex 4 cytochrome AA3 oxygen oxidoreductase. And next we are all going to ion sulfur center from prosthetic group of ion sulfur protein. Non-heme protein also function in the transfer of electron. For example, for FMNH2 to coenzyme Q and cytochrome B to cytochrome C1. Cytochrome Q10 is a fat soluble quinone compound. Cytochrome PCC1AA3 contain heme. Cytochrome AA3 also contain copper, and ATP synthetase is involved in the formation of ATP. And next we are all going to inhibitors. What are the inhibitors of electron transport chain? The complex 1 is inhibited by rotione and then amobarbital and pyridine A, and then complex 3 is inhibited by antimycin A and dimercaprol, and then complex 4 is inhibited by cyanide, hydrogen sulfide, carbon monoxide, sodium azide. And next we are all going to PO ratio. P means phosphate and O means oxygen. It is the number of high energy phosphate produced per atom of oxygen used, that is for NADH plus H plus ion. For FADH2, the NADH plus H plus ion give 2.5 ATP and FADH2 ion give 1.5 ATP. The respiratory control in the presence of adequate oxygens and substrate, ADP become rate limiting. And next we are all going to uncouplers of oxidative phosphorylation. The uncouplers are these substances that allow electrons transport chain to functions without phosphorylation, and so ATP is not synthesized but oxidation proceed. They are lipophilic and allow transport of H plus across the inner membrane but not through ATP synthetase, and so the proton gradient is cancelled without ATP formations and the free energy is liberated as heat. For example, 2,4-dinitrophenol. Thermogenin, that is uncoupling protein thyroxine, uncoupling protein thyroxine. And next we are all going to the adenine nucleotide transporter, ADP and ATP. The phosphate transporter, exchange of H2PO4 for OH, or co-transported. Co-transported, we are already see about co-transported of H2PO4 with H plus. The transport of reducing equivalents across the mitochondrial membrane from cystosol is mediated by two important shuttles. First one is malate and second one is malate and aspartate shuttle, and second one is flavoprotein dehydrogenase glycerol 3-phosphate shuttle. And next we are all going to substrate level phosphorylation. The formation of ATP within certain step of metabolic pathway, that is substrate level, without passing through electron transport chain, is called as substrate level phosphorylation. For example, pyruvate kinase, phosphoglycerate kinase, succinate thiokinase. Thank you, students. If you have any doubt about this topic, please comment to me. Thank you.